been great all through this. So today's talk is Chris Obara from Janelia. And one of the great things about this series is that we're meeting people who we wouldn't necessarily meet in our normal bubble. So I'm really looking forward to this. So just to give you a quick overview, Chris is a postdoc in Jennifer Lipkoff-Short's lab, and he's broadly interested in the way protein and lipid interactions have evolved to solve complex and energetic problems in living systems. So I think probably at that point I'll hand over to Chris because he can tell us more better himself.
Thank you so much, Alex. And thank you guys for inviting me for this. It's really an honor to get to speak in this forum. I'm going to share my screen here first. Good to go?
No, you're sharing the presenter view.
Oh, got to swap. All right.
Perfect.
Awesome. Okay. Yeah, so thank you again for inviting me to speak in this setting. I know that the more conventional way to give a talk in this forum is to introduce a new technology and sort of walk through the benefits of that technology. But I want to try to do something a little bit non-canonical today, which is tell you about a story that we've been working on. It's one of a few we've been doing in parallel, where we've tried to iterate back and forth between the advantages of high resolution but low specificity electron microscopy and the high specificity and dynamic nature of single molecule tracking. And sort of by going back and forth between these two tools, we've been able to parse something about a biological system that was previously inaccessible to us. So as you may be aware, the cytoplasm of eukaryotic cells is highly compartmentalized. This is a single slice through a high pressure frozen and freeze substituted electron microscopy volume of a COS7 cell grown on a cover slip. And what you can hopefully appreciate just looking at this with a conventional EM stain, which labels the membranes, is that this is very far from a wide open space. In fact, the vast majority of the cytosol is enclosed by membrane-bound organelles. And just as a reminder, conventional EM stains like this don't stain membraneless organelles very well, so it's actually even more crowded than what it seems like here. And this creates kind of an interesting space for cells to be able to navigate. Now, it turns out to be really important because organelles, although they are separate sealed subcompartments, still have to be able to synchronize their chemical reactions in a dynamic way for cells to be able to execute their functions. And the major way that this communication is thought to be maintained is at sites of specific contact between these compartments. Additionally, from a physics perspective, these sites of interface between organelles are of a lot of interest since they produce a very unique two-dimensional liquid environment. However, despite the fact that this communication must be very important and that we have, I think, had a lot of interest in it for some time, the structure and prevalence of this site is highly perturbed by conventional chemical fixation. In fact, if you compare high pressure frozen cells and chemically fixed cells in single slice EM, there's almost twice as many contact sites found in chemically fixed cells, suggesting that trying to study the nature of contact sites between organelles in a chemical fixed setting is probably looking for a needle in a haystack. However, we've been very fortunate over the last 5 years to be able to work in the era of high pressure freezing and collaborate heavily with Harold Hess's lab here at Janelia, who uses three-dimensional Fib-SEM to try to work in high pressure frozen and freeze substituted cells. And in this context, we've been able to reconstruct some contact sites like you can see here. And there's a few things that were immediately evident. First is that while they are indeed very prevalent, even in the absence of fixation artifacts, they actually are also heavily and closely associated with obvious biological phenomena. So you can see here just an example of one particular mitochondria where you can see the cristae of this mitochondria are all sort of lined up so that one of their ends at the mitochondrial membrane in a place where it's directly contacting the endoplasmic reticulum. And that's actually true for the vast majority of stacked cristae in tissue culture cells. So if this gives us some sense of what this interface looks like, it would probably be beneficial to understand something about the biological specificity with which this is executed. But whatever that specificity is, it must be incredibly dynamic, because our lab and others have published a number of papers over the last 5 years showing that contact sites between organelles are extremely dynamic in cells and can be broken or formed with timescales of just a few seconds. So fortunately, if we want to understand who confers this molecular specificity, we have a really long list of potential candidates from many years of very careful biochemistry, in which some of the genes you can see here for the ER and the mitochondria have been pulled out because biochemistry allows us to pull out mitochondrial associated membrane, i.e. fractions in a biochemical preparation that are associated with both organelles and are presumed to describe the interface. However, despite this long list of candidate proteins that we have, none of these proteins have been shown to be solely specifically at the contact site. And in fact, none of these sets of proteins are universally at contact sites in all cells or under all conditions, suggesting that even though contact sites themselves are very pervasive, the actual molecular players that are mediating them may be quite dynamic and change not only from cell type to cell type or in different tissue needs, but actually as a function of the specific needs of a cell at a given moment in time. And as you can imagine, this creates quite a challenge for people who would like to fluorescently label this system and understand something about what's going on, because by definition, any label targeted through one of these tools to the site will have background in at least one of the two compartments. And attempts to increase the signal to noise using things like crosslinkers, like fluorescence complementation or FRET assays, even at the low affinity of a FRET interaction, is actually sufficient to cause expansion and propagation of the contact sites, suggesting that these contact sites are very, very sensitive. It's a very fragile biophysical environment that's in perfect equilibrium, and even small amounts of perturbation can drive it awry. So our lab decided to take a more unconventional approach, I think, to understanding the system, in which we took advantage of the fact that a theoretical tether between the ER and the mitochondria is diffusing normally in the membrane of the ER, which is a much, much larger and pervasive membrane in the cell than the mitochondria. And so we wondered if we were to do very high-speed single molecule tracking of this ER tether. In this case, we chose the canonical tether VAPB, which is a pretty well-characterized ER to mitochondria tethering protein, that we might be able to identify the sites of contact solely as the place where the tether behaved unusually because it was bound to its mitochondrial binding partner. And so for this approach, we used the technique known as SPT-PALM, which was developed in the lab some years ago when Suliana Manley was a postdoc, in which individual fluorophores are photoconverted at very low density. You can see here an example, just a few at a time in a field of view to avoid linkage errors. And then this is collected at very high speed video rate microscopy to be able to form trajectories of where molecules move during time. Now, while this tool has been around for some time, these trajectories that are coming out cannot be directly interpreted by conventional statistical physics approaches that are used for single particle tracking, because these molecules are not freely diffusing in an isotropic system, but rather are confined to the endoplasmic reticulum. And the confinement to the organelle has such a dominating effect on the mean squared displacements that things like MSD fits do not work. So to fix this problem, what we've done is, over the last 5 years, worked out a relatively careful system in which we use a simple dichroic mirror to collect simultaneous information about where the underlying organelle is. In this context, you can see that this means these trajectories are completely contained within the tubular endoplasmic reticulum in the periphery of the cell. And although I'm not going to explain all the details of it today, I'm happy to address more about questions at the end if you're curious. We've worked with 3 different theoretical physicists to develop 3 different implementations for how we can appropriately correct the boundary conditions for traditional statistical physics approaches in single particle tracking and pull signal back out of the noise of the system. And thankfully, in giving us confidence in the way that we've done this, all 3 of those qualitatively converge for the vast majority of biological processes, and 2 of them even quantitatively converge for diffusion. And so we have some faith in our ability to pull signal from this. So if you apply this type of approach to something like a simple tail anchor, where we've simply targeted the fluorophore to the ER membrane and allowed it to diffuse freely, what you can see is that you collect trajectories of single molecules moving around in the ER, and the trajectories are essentially completely contained within the endoplasmic reticulum. You can see they explore all of the space. And in places where they show more complexity, it's because the ER itself also shows more complexity in those spaces. However, if you do this with this canonical ER to mitochondrial tethering protein, VAPB, you start to see lots of regions of putative interaction, as visualized by very tightly clustered trajectories that we've indicated with these black arrows here. This is most easily visualized or quantified by simply looking at the probability of finding a molecule within a defined time window. And when you perform this type of an analysis, you can derive probabilities of molecular localization over time. And what you see is that in a conventional tail anchor, while this probability is uniformly distributed over the structure throughout space and time, in the ER to mitochondria tethering protein VAPB, you get the transient formation of hotspots as individual tethers are formed. And as you might expect, since this is an ER to mitochondria tethering protein, the vast majority of these co-localize with the mitochondria if you add an additional channel and image where those are. In fact, it's sufficiently correlated that you can see that during the time of a very short movie, in this case 1 minute, every place that these two organelles touched one another, you get a transient hotspot in tether behavior as the individual molecular tethers rush into the space and try to stabilize the interaction as it forms. One thing that is a nice sanity check is that this molecule VAPB has also been implicated in binding, admittedly at lower propensity, but with some interaction with other organelles like endosomes and Golgi. And of course, in these cells, we also see contact site formation in spots where there are no mitochondria, giving us confidence again that we're actually looking at what we wanted to see. And just to validate that this is indeed contact sites that we're looking at, we went back to the 3D electron microscopy data that we had before, and we modeled diffusion on the surface. And when that modeling is performed, we can convolve the data that we get out of it as a result of the point spread function of our optical microscope, which we've of course measured very carefully. And doing this, we can generate theoretical contact site visualization in the projection plane. And what we essentially see is that when we do this from the 3D EM, high pressure frozen data, and when we do this from experimental data collected on the microscope, qualitatively, we see something that looks very similar where contact sites are visible. And if you quantify the size of these subject to the lower resolution of single particle tracking compared to EM, essentially these distributions are indistinguishable. So I think, again, suggesting that we're actually looking at contact sites. As a last sanity check, we simply went back and deleted the interacting domain of this protein to make sure that when it was not able to interact with the mitochondria, we lost the contact sites. And again, indeed, you can see in this situation that the contact sites are gone. And if you fuse the interacting domain of this protein back to a non-specific ER-targeted transmembrane protein, you actually regenerate the presence of the contact sites, they come back. So at this point, I think this tool for looking at contact sites is an effective way to sort of visualize them in what's hopefully a non-perturbative way. However, although we've now generated a super resolved probability-based image of what contact sites look like between the two organelles, we actually have more information because each of these contact sites is assembled from individual molecular trajectories that are distinct, and we know what happened to molecules up and downstream of their interaction with the contact site. So you can see here is a single trajectory that was diffusing through the ER, entered the contact site, and then left the contact site again. And the behavior that it had at each of these points can be easily parsed with a simple Bayesian tool. So we've simply colored the trajectory here with the red being the portion in the contact site and the blue being the portions where it's freely diffusing in the ER. And if you break these trajectories apart across many contact sites in many cells, what you can see is there's a significant reduction in the diffusion within the contact site, as you might expect, since the molecule is bound to its binding partner there. And in fact, if you just take segments of trajectories in these two different states and plot them with their center at an origin, what you can hopefully see is that there's a very different biophysical landscape that molecules experience when they're freely diffusing, where they can travel over long distances, as opposed to when they sit confined in a contact site, which is shown in these red trajectories on the right. However, we can actually take this one step further because although some contact sites are quite short-lived, only lasting for a few seconds, there are many contact sites that are stable even over a time course of minutes. And during the course of a minute or two movie, many hundreds of individual molecules will explore these spaces. So we can take all of those individual trajectories and break them apart based on what happened to each molecule when it was at a different subregion of the contact site. We do this functionally by splitting the contact site into Voronoi tessellations. But what you end up with is something that looks like this, where here are two contact sites that are directly next to each other on the same mitochondrion. And what we've done is simply mapped the diffusional landscape inside these two contact sites as a function of the sum of all the individual molecules that explored that space. And one thing that came out of this that was quite surprising is that actually not all contact sites have the same diffusional speed. In fact, these two contact sites are right next to each other on the same organelle, but you can see that the one on the left has much slower diffusion occurring in it than the one on the right. That said, there are some similarities. So if you just draw line plots across these two and sort of plot the diffusional speed itself, the coloring in the back of these graphs is the molecular density. So you can see the density is going up as the diffusion goes down, which makes sense where the two tethers are becoming in higher density at the center of the contact site. But what you can see is that all these contact sites, and this is true for all of the hundreds of contact sites in more than 25 cells across many different conditions, all of them have in common a sort of central structure where there's a low diffusional well at the center of the contact site, and then a gradual decrease in the restriction of diffusion as you move towards the edges. So we wondered if we might be able to go back to our 3D EM data set and look for something that might give us some insight into how this is mediated. So here is just an example of a single contact site on a single tubule. I've selected this one because you don't have a lot of extra entry points from the ER that you would get with a more complex structure. But if I remove the mitochondria and color code the ER membrane by its curvature, there's something quite notable about this structure, which is unlike the entirety of the rest of the organelle, the ER is uniformly very highly positively curved because it's such made up of such tiny tubules. However, in the region at the center of the contact site with the mitochondria, it actually takes on a high degree of negative curvature as it's taking on sort of the imprint of the mitochondria where it is bound. However, there's sort of a functional consequence for this, which is that if I'm a single tethering molecule coming through the ER membrane and I come into this contact site, I by definition have to pass through this red region, which is a region where the two membranes are close enough for binding to occur. Tethering can happen here, but there's not sufficient density of molecular tethers within this space to pull the two membranes into direct parallel apposition as you get in the center of the contact site. And so what you might imagine is that as new tethers come into this space, they can reach their binding partner, and then they're in a situation where these two organelles are moving around in the cell, and the membranes here are not closely supported. And so it's energetically favorable then for the molecule to slide laterally into the center of the contact site where the molecular density is high enough to hold the two membranes at the right distance. And so we think this might be something about how new tethers are formed. It also carries important implications for how contact sites can be broken, since dynamic molecules in this space would then sort of lose their association and then diffuse, no longer have this energetic favorability for the contact site, and would be able to diffuse freely out into the rest of the structure. And it turns out, in fact, if you look at the single molecular trajectories, you can see evidence of this. So what I've done here is we've just taken many individual contact sites that are present at the periphery of cells on individual tubules where the structure is non-confounding, and we have lined up the entry points of all of these contact sites on the right side of this graph here. And so if you just choose a line that I've drawn here, in this case arbitrarily chosen to be at 90 degrees, and look at the probability distribution of molecules within these contact sites when they're all put together, you can see that it follows a Gaussian distribution as you might expect. However, if I swing this line around and draw it in the direction of the entry, what you can see is that actually this Gaussian picks up a large degree of probability shoulder where there is molecular localization and recruitment, but not at the same propensity as you get towards the center of the contact site. And so what we think this actually is representative of is this red region that I indicated to you before. These are the places where molecules can interact, and so they're slowed and they're there with more propensity, but they have not shifted into sort of this high density well where the two membranes have now really taken on one another's structure. And we hope that this kind of approach can allow us to begin to start to understand something about how contact sites are regulated at the level of the single molecular players using some modeling approaches in the future. One interesting thing about this interface, though, is that this brings up an interesting problem for cells, which is illustrated in this image here. You can see here we've got mitochondria colored in blue, and the ER is colored in this black color. And there is a lot more ER membrane in a cell than there is mitochondrial membrane. In fact, there is more ER membrane in cells than there is all the other membranes in the cells added together, including the plasma membrane. And so this creates a complicated situation for cells, because unless there is a vastly larger amount of the ER tethering molecule in the cell, there will always be shortages of the amount of ER tether available for a binding event to occur, because there's simply so much more space for those molecules to have to explore before they find the mitochondria. And so we wondered how this might be regulated, and if we might be able to understand something about the way that regulation would happen based on which of these two players was involved. And so the experiment we did is really simple. We simply took control cells where we overexpressed an abundance of the ER tether, in this case VAPB, or where we overexpressed an abundance of the binding factor in the mitochondria, PTP-IP51. And what you can hopefully appreciate here is that there is essentially no significant changes to the structure of the ER of the mitochondria or their interface when we only overexpress the ER factor, suggesting that this interaction is indeed governed by mass action to some extent. But this interface is highly, highly sensitive to the expression level of the mitochondria factor, since there's far less mitochondrial membrane. And even with a very slight amount of overexpression, you get this really dramatic restructuring in which the ER essentially wraps the mitochondria like a little burrito and seals it into these sealed subcompartments. We then went back and looked at trajectories in this situation to try to understand what happened. And as you might expect, contact sites in this situation are expanded. They show a significant increase in size as a result of the fact that there is now more binding factor available. And so the contact site is sort of just spread. If you quantify that, you can see that that happens just with a simple ellipse fit. It happens both along the major and minor axis. However, one thing that's notable is although the contact sites are much larger and expanded in this system, they actually are not, there's not more of them. So you don't generate new contact sites by overexpressing the mitochondrial binding factor. The contact sites are already there. They just spread outwards to fill the space. One thing that was notable, though, is in overexpression of the mitochondrial binding factor, you do essentially completely deplete non-mitochondrial contact sites from the cell, suggesting that, again, this mass action, the law of mass action is governing this interaction. So there's a very high dependence on the concentration of the mitochondrial binding factor. And eventually, if you were to drive the expression really high, you might be able to deplete the free VAPB pool. And so just to prove that principle, what we did is we overexpressed both VAPB and the mitochondrial binding factor, PTIP51. What you're looking at here are only ER proteins. We have no mitochondrial label in this cell whatsoever. But you can see that the ER has wrapped so closely around the mitochondria that you can actually make out where all the mitochondria are in the cell very clearly. And if you just look at a non-specific Sec61 beta as a non-specific ER targeting motif for the fluorophore, it does, of course, label this region of ER very avidly, but it also is labeling the peripheral tubules. However, if you look at the VAPB, even in the overexpressed context, you can see that actually it's essentially completely depleted from the peripheral ER as these contact sites have just swollen to hold it. So we wondered if maybe we could think about a situation where this might happen that's biologically relevant in cells. Now, there's a significant amount of literature suggesting by single slice EM that contact sites should expand or become more avid in the context of a nutrient deprivation. And this is because the ER signals to the mitochondria to activate the OXPHOS pathway and feed the cell. And indeed, if we look in this system, we can see that the contact sites do become bigger when cells are transiently deprived of nutrients. And so you can see here, we've just fit sort of minor and major axes to each contact site, and you can see that these expand. One thing that's notable is that under this condition, while there is an expansion in both axes for the contact sites, they do tend to elongate. So actually the major axis has become larger by a larger factor than the minor axis, suggesting sort of the contact site is sort of zippering outwards. We think this sort of makes sense based on the structure that we have. If you imagine new tethers coming into this space and causing the contact site to sort of extend along the mitochondria, it's going to be easier to do this along the dimension that both the ER and the mitochondria exist, and you sort of get elongated contact sites in that context. One thing that's notable, though, is although the contact sites do get significantly larger in this context, they also have some physical changes in their dynamics. So you can see here, we've just mapped the relative diffusion of a representative contact site in a normal cell versus one that's been nutrient deprived. And you can see there's a significant reduction in the diffusion coefficient within the contact site in the nutrient deprived cell. And you can sort of see that more clearly here, where I've just drawn lines through these two contact sites to map what this looks like. So contact sites are not only expanding, they're also becoming associated diffusion coefficient. I'd like to talk about another example that we looked at that has turned out to be quite interesting, which is that this molecule VAPB has a disease mutation in it, the P56S mutation, which is heavily linked to ALS. This is a penetrant, highly dominant mutation that causes ALS in humans. And many years of work have demonstrated via biochemistry that it seems the vast majority of P56S VAPB is in an insoluble aggregated form in the cytosol, and probably as a result of early misfolding. However, it remains a major question in the field how these aggregates contribute to pathogenesis, because the correlate of ALS-induced disease in neurons is actually an elevated ER to mitochondrial communication phenotype, not a reduced one. And so to try to understand this, we thought, well, maybe we could use single particle tracking. And so we started doing single molecule tracking of VAPB molecules with this P56S mutation in them. And I'm just showing you some representative trajectories here. Indeed, the vast majority of the trajectories are in an insoluble aggregated form. I've showed you a few examples of them here. They're color-coded in black, so you can see what those look like. However, we'd like to point out there are still a significant number that do effectively get inserted into the ER, and these diffuse freely in the ER, as coded here in blue, very similarly to the way that wild-type VAPB does, as you might expect. However, there's also a subset, and this is admittedly a small fraction of the total number of molecules, but there's a subset that we've color-coded here in red, which show what is clearly still a contact site-associated phenotype. And in fact, 100% of these are still at the mitochondria. And so if we just pull these three species out and show you segments that are associated with these, you get some sense that, again, the freely diffusing ones look a lot like the wild type did before. But if we blow up on these contact site associated ones, you can see there are some differences between the P56S at the contact and the aggregated forms. However, these P56 at the contact actually look more similar to the aggregated forms than they do to the freely diffusing ones, or sorry, contact site associated diffusing ones that we saw in the wild type condition. And so we wondered, is there something about this behavior that we might be able to see in the contact sites themselves? Is something about these contact sites perturbed? So the first thing we did is exactly what I showed you before. We look at the size and shape of these contact sites, and as you might expect, maybe there is essentially no difference. These contact sites are exactly the same size, they have the same shape, they have the same abundance. It doesn't seem to be any obvious mistake in terms of the simple contact site shape or distribution. However, when we looked at the diffusion coefficient, we see a very slight reduction. Nothing nearly as dramatic as what you see with the starvation condition, but there is a slight reduction, and we wondered if there might be something to this. And so we went in and looked at this diffusional landscape. And this is one of these moments in life when the fact that I'm OCD and insist on looking at every single piece of data too many times turned out to be useful. So if you look at this diffusional landscape, what you can see is in wild type cells, I showed you before, all of them have a nice sort of symmetrical single diffusional well that is sort of represented here on the left. However, when you look at the P56S contact sites, what you often see are sort of these weird, like slightly asymmetric wells. And we wondered if there might be something that could explain this in the single molecular trajectories. It took us some time to figure out exactly what the problem was here, but it turns out that if you look at a single molecular trajectory in a single contact site, which is what I'm showing you here, for every single one of the many hundreds of contact sites we looked at, more than thousands of trajectories across many, many cells, during the time that the molecule moves around, it explores the entire contact site that's open to it. So I've traced the edge of the contact site in this dotted black line here, and you can see these single VAPB molecules that move in there explore that entire space promiscuously. However, in the P56S condition, more than 70% of the single trajectories in P56S contact sites only explored a small subset of the contact site and didn't actually explore the whole space, suggesting that there is a lack of fluidity within that system in the P56S condition. And once we knew that this is what we should be looking for, we went and looked for the rare example of a contact site that forms at the periphery of cells where it's very stretched out, maybe a tubule and a mitochondria that are perfectly parallel right at the edge. And indeed, when you look in P56S under these conditions, you can see this contact site is nice and long, so everything is spatially distributed evenly. You can see that there's actually not a single well in this contact site like normal wild type contact sites have, but in fact, 3 separate wells that formed. And indeed, each of these is associated with the places that these molecules are getting stuck. So whether that has to do with a decreased diffusional landscape or the P56S is actually aggregating inside the contact sites themselves, we're not sure. But one important thing that would come from this is you could imagine that if these contact sites have to be dynamically reassembled and then disassembled over a second to second time scale, having any form of sort of aggregation or inaccessible regions of the contact site would make the disassembly of this contact site quite challenging. And since contact sites give positive signals in general to mitochondria to activate them, we think this might carry some implications for the disease phenotype. Even though the vast majority of the P56S molecules are aggregated in the cytosol, it may be the very small number that get into a very sensitive piece of machinery, the contact site, and sort of jam it up that actually confer the pathogenic capacity of this genetic mutation. So with that, I'll start concluding and say that the interface between organelles can be a highly plastic and dynamic environment. And this VAPB diffusional landscape that we've mapped for you today is highly variable, but it does seem to correlate to local ER curvature, suggesting that tethering molecules and the shape of the membrane are intimately connected to one another, as you might expect. Contact sites do expand dynamically under nutrient deprivation or when the binding factor is overexpressed in the mitochondria, and that interaction is governed by mass action. And that pathogenic mutations in VAPB can cause an altered dynamic landscape within these contact sites, which may underlie an altered ability to break or reduce them, hence leading to disease. So I think this is just the beginning of a way to start thinking about this really difficult to study compartment in cells. We've got, I think, a lot of exciting ideas about ways to look at other tethering molecules and how they change. And I'd really like to, if anyone is interested in modeling, let me know. I think we'd like to try to model something about how this could be governed and what the effects would be on the structure, just from principles, now that we have some sense of how this works. I'd also like to end with sort of just like a shameless plug for this type of correlative single particle tracking approach, because even though I've showed you today about interactions with an organelle, actually you can see lots of other kinds of behavior with this tool. And we've worked with a really talented grad student, Tim Obel at the MDC, to work out 2-color tracking in the system. And you can see here, these are 2 proteins that are directly bound to one another. And we know that they're interacting or part of the same complex because they move in perfect correlated motion over an extended period of time. We can see when molecules bind to microtubules or cytoskeletal elements because they become essentially completely immobilized. And that is very distinctive from a molecule that is freely diffusing. This molecule here on the right is the identical molecule to the one I just showed you here in the center, but we've mutated out the binding domain. And now, of course, it's able to freely diffuse through the network with no obvious inhibitions. And so we hope that this kind of approach might be able to help us understand something about really doing biophysical assays inside of living cells, rather than having to stick to in vitro systems or work solely on the plasma membrane or in the nucleoplasm. With that, I'd like to thank, this is my lab and our group at Janelia. It's gotten quite a bit smaller over the last few years. Janelia has pretty small group size. And all of this project I showed you today about VAPB was a collaboration with Johnny Nixon Abel. He was a postdoc in Craig Blackstone's lab now some years ago. Obviously, I need to thank the theoretical physicists who helped us build this whole framework, Chris and Jean-Baptiste, and this whole group here on the right, the Betzig and Hess labs, who helped us work out the correlative system for getting the 3D-EM working the way we needed and being able to understand something about our system. So thank you very much, and I will be happy to take questions.
Right. Thank you very much for such an interesting talk, Chris. Before we run into the questions, we traditionally do the quiz, so I'm going to set that off. So I'll start that sharing now. Here we go. Anybody else who wants to, I mean, so obviously the link was in the chat in the talk on the sidebar, if you can see that. No, we've got no participants, come on. Let me see if I can switch back. But anyway, the link was in the chat there. Oh, here we go. Some people are coming in. So we'll wait a little bit longer. And everybody else, if you got some questions, please feel free to type them into the chat, and we'll get to them in a minute. Otherwise, and if you're still there, I'll try and highlight you so you can ask them in question. But,
can you put it again, the link? Because if you came later, I don't think it's visible.
Let me go back. Let me see what I can do. Here we go.
Just in the chat, if you put it.
I can't get back to mention.
Okay, that's fine.
It's definitely in the chat. All right, we've got 4 people on board so far. Let's see what happens. No, I can't get back onto that. Without stopping. Always least. Okay, let's see if we get another. Oh, we've got 3 more participants. Oh, yeah, here we go. All right, so you get to win a fantastic fold scope. What more enticement can I offer? Okay, we're getting quite a few suggestions on what we next tether the key to. So, all right, how about we start the quiz? So let's just go. So remember the same thing. The quicker you can answer, the more points you get. And here we go. Question 1. Are you ready? A major factor complicating the study of mammalian organelle contact sites is, oh my goodness me, electron microscopy cannot be done in live. You have to read these yourself fast so I can say them out. And all of the above is option number E. And we got 4 and 4. Okay, we got 4 good answers there. Okay, some people have got points. Let's see what we've got. Oh, CVS is winning. Okay, great. So and GF is right on the tail. So let's see what happens in the next question. Question number 2. Yeah, I have to apologize. I can't do anything about the font size on these questions. Traditional fitting of mean square displacement-based models for single particle tracking does not work well in the internal organelles because MSD fits are not linear, imaging speeds are not fast enough to model it in such quick motion, confining of the organelle dominates. Okay, got some answers. And answer 3, confinement was the actual correct answer there. Let's see if the stats have changed at all. All changed. Vroom, I think, is now the winner. Let's see where we go. Yep. And GF is number 2 still. Okay, so. All right, question 3. One control that becomes especially important in single particle tracking within confusing structures is a test to prove that linking errors in the data are minimal, checking for uneven blinking rates across the structure, test laser pulse speed, checking the microscope stage is actually hot and temperature, or none of the above. And none of the above. And the correct answer is a test to prove that linking errors in the data are minimal. Okay, let's see where that puts us. You've given us a really hard quiz this week, Chris. But never mind, we got some real contenders.
I'm sorry.
That's absolutely fine. Just a minute. Did that update? That didn't update. Well, I can't force it to. I've only got a forward button, not a backward Okay, here we go.
I think only one person got it right, so it probably did update.
Ah, yes, yes. Good thinking. I'm glad you're ahead of me. Okay. So we can get the next question. Correlating high-speed single particle tracking to underlying structure requires. No, I know you definitely gave the right answer to this one because I heard it. A fancy home-built microscope, a powerful computing cluster, a dichroic mirror to split the channels, SCMOS cameras, or a long working distance objective. Yeah, everyone was listening. Well, a lot of people were listening. Okay, let's see where that takes. Now we should get an update on the leaderboard. Wow, Vroom. Oh, and Sarah is right on your tail, Vroom. So let's see what happens on the final question. Okay. Right. A single point mutation in VAPB has the following effects on mammalian contact sites: increased contact site size, in contact site diffusion speed, increased tether speed, changes in the main axis ratio, or none of the above. And again, I know you don't answer. Yes. And one, oh, this might be the decider. Then let's see. CRUK got that one, I think. I think that then leaves the Vroom is the unchallenged winner. Okay, Vroom, if you could identify yourself in the chat, that would be great. You can just send your email to the RMS. We will send you that fold scope. Okay, so we have a few questions in the chat. I think let's start with Victoria. Do you want to unmute yourself and ask your question directly to Chris? Victoria,
sure, yeah. Can you hear me?
Yep.
Hi, Chris. That was a fantastic talk. Thank you so much. I just wanted to ask, in this case of the ALS that you see the P56S mutation, do you see also more tangles in between the mitochondria and ER as well?
So if you put this into a tissue culture cell, you do get tangles in that interface, and you don't at all in wild type cells. So it only happens with the P56S mutation.
Yeah.
But we've never done this in like a patient sample or in neurons, just because introducing the labels and stuff is quite complicated. So I don't know to what extent this will, if you were willing to buy a tissue culture cell as a decent model for this, then yeah, this is something important to think about. But I don't know,
Yeah, I see. Okay, thank you so much.
All right, great. And then Wei Ching, if you want to unmute, and you can ask if you're still there. Wei Ching, okay.
Hang on, sorry.
Here we go. Fire away.
I've got 3 questions. Sorry, I'm a bit greedy.
That's
and they're
all.
all quite technical. One is how many, I presume you can count, because it's single molecule, I presume you can count the number of VAPB at these ER-mitochondrial contact site. Do you have a tally of that?
Yeah, so you can, but it's not as simple as a fixed cell single molecule experiment where you can sample to exhaustion, right? So you're doing this all live. So I can give you a relative count for how many molecules you sampled, but that's probably significantly less than the total number of molecules who are there. And there's no way to strap that back with our current technology. So, I mean, the average contact site in our data collects somewhere in the range of like between 30 and 120 molecular trajectories. I'm sure there are more molecules since we're only turning 1 or 2 on at a time, right? It's probably quite a bit more than that in there.
Okay, and I probably, you blink over your graph too fast, and you must have a draw. What's the average draw time of VAPB at these contacts?
Yeah, this is a really good question. We don't have a good estimate of that because the molecular, so the lifetime of the fluorophore is a distribution that peaks at 3 seconds, and you get some that last as long as 10 or 12, but like the vast majority are less than that. The dwell time is somewhere in the range of a few seconds, somewhere between more than 3, but less than 12. But I don't have a good way to estimate that number with much confidence.
Wow, that's super transient. So this will be super difficult to catch on a fixed cell.
Yeah.
Or pull down or anything.
Yeah. And I think this is why people have had such a hard time visualizing this. I mean, amongst other things, right, in things like confocal microscopy, because it's a very slight increase in the expression level, right, as a result of these delayed interactions.
Holy cow. Okay, and my third one is, okay, this is just pure experimental thing. How did you make your nutrient starvation condition?
Yeah, good question. It was a sledgehammer. So we just put the cells in HBSS for 8 hours. So they're real hungry.
Have you tried, say, like a galactose or like glucose starvation?
We haven't done any specific starvation conditions. I think this would be really interesting. So one thing that I've been itching to do for years, and we haven't done it yet, so disclaimed, this setup with the dichromatics to split fluorescence, is compatible with any color, right? So what I'd really love to do is be able to track molecular tethers while also quantifying membrane potential in the mitochondria, right?
Oh, wow.
So you can see as oxphos goes up, right, are these tether, like, who goes first, right? Are you tethering first, then calcium, that, I think that's probably the system you want to do that experiment in, because you'll be able to pull all of the subtleties, right, of the response apart. Yeah, we haven't done it yet. If someone is interested or has an idea or has a reagent you want to try, let us know. We're all game to give it a shot. We just haven't entered that space yet.
All right, thank you very much, Wei-Ching. And Cynthia, are you there? Do you want to unmute and ask your question in person? Cynthia. No? Okay, well, okay, I'll just ask it for you. Wonderful talk, so that's from Cynthia. Thank you. 2 questions. What are the fluorophores used, and how many points do you have in a good trajectory? Actually, I was going to ask that, but so what fluorophores and what's your...
Yeah, sorry, I should have said that. So everything we're doing here is tracking Janelia floor dyes that are linked to a halo tag. Everything I've shown you here today is a conventional Janelia floor dye, so it's photoactivatable, but it's conventional PA646. There are new generations of these dyes that are significantly better and go longer, but I'm not redoing everything with them now. That's the way we do it. And I would say, so for us, it depends on the analysis. So I kind of flip the cards around here. There's 3 different analysis pipelines that we use to analyze the data, and different ones have different requirements for how long a trajectory would be. So none of our data that I showed you today has a trajectory with less than 100 steps. So everything is cut at that point because you avoid noise that way. However, the more fancy analysis, like the Bayesian state splitting, like the way we were able to identify the P56S states, that requires significantly more. And so for those, we don't do any trajectories that have less than 500 steps.
Okay, so actually that's a lot more than you get out of any of the traditional PA fluorescent proteins, isn't it?
Right.
Yeah.
Right. The other problem with fluorescent proteins is that they blink, right? And so the same molecule comes back on and off a lot. These JF dyes we're tracking have been designed to minimize blinking. So the average molecule blinks, 1 or 2 times like total, and then it's done. So it really decreases your problem with the same molecule turning back on later and then messing up your analysis.
Okay, we've got another question from Sarah Schweighofer. Sorry if I got your name wrong. If you're there, please feel free to unmute and ask your question. Put your camera on if you're there. Sarah, you want to have a go? Okay. All right, well, I can answer it. Anyway, again, really cool work. That's thanks from Sarah. A couple of technical questions. Okay, sort of the thing. How long can you measure one trajectory? Oh, I guess that's 100 or 500 steps. What was your frame rate?
So ours is 100 frames a second is everything I showed you today. So yeah, cutoff would be either whatever that is, either 1 second or 5 seconds for the 2 analysis pipelines. The longest trajectories we get will maybe last 10 to 12 seconds, but I think those are very rare.
So, I mean, I know you stressed this, but how exact is the overlay with the, I guess it's wide field ER network that you're sort of...
Yeah, no, this is a good question. So it depends again on the analysis pipeline we're using. So raw overlay is done with fiducials, right, to put the two things on top of each other, and that's like maybe pixel level resolution. For some of these statistical tools where you want to unwrap the structure, we have to be much finer than that. And so we have a system where we basically run a kernel along the structure, fit the center point with conventional centroid fitting, and then use that to refine the center axis and then put the trajectories back onto that.
Right, okay. Let's see if there are any more questions. Oh yeah, Sarah says, sorry, no mic. Oh, actually, Sarah, just as a follow-up, is there a publication for this work?
Ask me in 3 weeks, maybe.
Okay. If you've got a bioarchive preprint, you know, feel free to stick it in the chat there. Okay, so just a minute. Apart from general expressions of thanks, Alex is saying, feel free to watch the rest of the Imaging One World Talks in the YouTube channel. Actually, I had a question for you. So how much of your work is sort of restricted out into the peripheral ER versus some of the central stuff? And I guess that's sort of reflecting on the three-dimensional nature of this.
Yeah, for sure. Everything I've shown you today is in the peripheral ER for exactly that reason. So we're not accounting for the third dimension. If you use a PSF engineering tool, you can go to 3 dimensions, but thus far we have been unable to achieve sufficient resolution in the third dimension to be useful in the ER. So if you want to track the mitochondria inside the cell, you can do that with a PSF engineering situation because, you know, 400 nanometers from the top to the bottom is enough. You can distinguish them. But these ER tubules go down to, you know, sub 50 nanometers. So confidently resolving the top and the bottom of the tubule is beneath the resolution that we have with this tool. I think there's some really interesting ideas of ways this might be improved going forwards. There's someone here in the building who's trying to work on an interferometry-based single particle tracking setup, which would of course be amazing if it works. But as I'm sure many of you know, interferometry is never an easy thing to make work reliably. So I'm optimistic, but don't want to promise you anything yet.
Okay, well that's a good answer. All right, so unless there are more questions, I'll just have a quick scroll through here and see if I can see some more. What I will do while I'm waffling on is just to remind you, next week we've got Rita Strack from Nature Methods, who will be talking about publishing methodological developments in imaging and microscopy in Nature Methods. General thanks to you from the chat as well, Chris. So I mean, I would like just maybe extend the thanks from the Imaging One World team and from the Royal Microscopical Society and all of our audience. So thank you so much for getting up very early to give us such a great talk.
Thank you so much for having me, and thanks for the questions. They were awesome.
Okay, all right. Well, in that case, I think we'll just draw today's work for the meeting to a conclusion. I'll just give you an awkward clap from on behalf of the rest of the audience. Thank you very much. There you go.
Thank you so much, guys. I appreciate it.
Okay.
Thank you.